All right. Welcome, everybody, to Standing for Truth. My name is Donny Budinsky, and I am going to be giving a presentation here in this video answering comprehensively the so-called best scientific evidence for evolution or common descent. This is the definitive talk on endogenous retroviruses. And let's get right into it. We have a lot to cover here on the topic of ERVs. Again, the so-called best evidence for evolution. And what I mean by the definitive talk on endogenous retroviruses is I am going to leave no stone unturned in this technical presentation, absolutely dismantling this argument. If you search in YouTube or Google the best evidence for evolution, you will see endogenous retroviruses come up from many channels that lean evolution or promote heavily the topic of evolution. And some have even said that endogenous retroviruses are as close to proof that you can get for something. And so what we like to do here at Standing for Truth Ministries is debunk their best challenges, their best arguments. And that is why in this presentation, addressing endogenous retroviruses, it is going to be thorough. We are going to be covering the details. I am going to be going over exactly why the creation model better explains the existence of shared ERV and ERV-like sequences in the biological world. And I am going to cover the superior model, as we don't want to just refute or counter evidence for evolution. What we want to do is model build. Model building is important. And so, yes, endogenous retroviruses are not good evidence for evolution. But in this presentation, we are also going to offer you a superior model. And so let's get into it. This is presented by myself, Donny Budinsky, founder here of Standing for Truth Ministries. We seek to address all challenges and objections to biblical creation. You can find endless content. This includes answering challenges, engaging objections, and answering questions in general, really on just about every topic. Of course, origins, ancestry, creation, evolution, but also on topics dealing with theological issues, soteriology, eschatology. And so please do make sure to take advantage of our website here, standingfortruthministries.com, and search through the video section. Especially, you can scroll down in the video section and see where it says guests on Standing for Truth, and you'll see all of the experts, the visiting scholars, the creation scientists that have been on the program. And you can see all the different subjects and topics ranging from biology to geology to genetics, astronomy, just about everything. Now, I have written an extensive book on the topic of endogenous retroviruses. This is the Endogenous Retrovirus Handbook. I have it here in front of me, dismantling the best evidence for common descent. It is a comprehensive dismantling of ERVs, and I'm excited to say that I am working on very diligently an expanded and updated edition where I cover in more detail the superior model. In the updated version, I will be addressing what the critics have said, how the critics have responded to my book and my arguments, over the last 2 to 3 years. And the follow-up, which will be an expanded edition, it will be even more comprehensive. Therefore, stay tuned for that. The current version is available on Amazon. Why is this topic important? Well, I briefly covered why it is important in my introduction. It is because as creationists, as Christians, we do want to engage. We want to answer objections and challenges to the faith, to what the Bible has to say. And we also want to counter pseudoscientific ideas. We want to uphold good empirical science, the kind of science that is observable, testable, repeatable, and truth. Truth is important. That's why our ministry is called Standing for Truth, Standing for Truth Ministries. Now, if we are going to talk creation and uphold the truth of biblical creation, then what we don't want to do is ignore challenges, ignore objections, pretend like the critics don't exist, or pretend like those who believe in evolution and reject creation don't exist. So we want to see what the best they have to offer is, which is endogenous retroviruses. And we want to engage their best in a sophisticated and in a comprehensive manner. And so that's what we're going to do here in this presentation. Now, let's make ERVs fun again. I don't know how much fun ERVs have been. I don't know how much fun ERVs can be. It is a technical topic. It is in many ways a hyper specific topic, but an important topic, because if this is the issue that so many have said, one, is the best evidence for evolution, and two, those that are on the fence, there have been many who are on the fence in terms of creation or evolution, but they are waiting for an adequate or sufficient answer to endogenous retroviruses, then what I want to do is make this topic as fun as possible. It is technical. The details are important. We are going to be explaining and going over the details because it's important that we accurately and appropriately represent the other side, the evolution, the common descent side, before we work towards countering their arguments. We want to understand where they're coming from. Why do they actually believe that shared endogenous retroviral-like sequences in the biological world are good and, according to many, irrefutable evidence for common descent? So let's make endogenous retroviruses fun. Again, the important question that I want you guys to consider, that I want you guys to ask yourselves as I'm going through this presentation is this: are endogenous retroviruses really the ancient remnants of past viral infections, or are they created units of DNA function? This right here is an incredibly important point that I want you to consider throughout this technical lecture. And the reason why is because the creation model and the evolution model, they have in many ways opposite expectations and contradictory predictions. The evolutionary community, evolutionary scientists, would largely predict that what we see in the genomes of living organisms, let's take human beings, for example, is a reflection of deep evolutionary history. Mutations have been accumulating for millions of years. Viruses have been infecting biological organisms over the course of millions of years. We've been accumulating genetic errors and genetic mistakes. And so what they would expect to find in genomes are a reflection of that. Therefore, they expect to find a lot of genetic mistakes, shared genetic mistakes, fossil viruses, evidence of evolutionary leftovers or evolutionary junk. But creationists, from the design perspective, from the design starting point, we expect treasure, not junk. We expect evidence for design. We expect evidence for special creation. We expect evidence for function. And that's why this question here is important. Now, here are important questions that we are going to answer and answer sufficiently, plus a teaser. Why are ERV-like sequences shared between the genomes of organisms? That's a major question. That's a major point that the evolutionary community likes to focus on. Shared endogenous retroviral-like sequences between the genomes of different organisms, to them, point back to common ancestry. Teaser, the answer is common design. It is like asking the question, why do all modes of transportation share engines in common? Why do they have batteries in common? Why do they have windshields in common? Why do a lot of vehicles have 4 tires? Why do they have a spare tire, a jack, backup cameras? Why do they have steering wheels? Okay. And the evolutionary community likes to say, well, cars don't reproduce. Well, for one, that's a misunderstanding of the design model, which we'll get into. And secondly, adding reproduction adds more complexity. It makes the problem more difficult. It makes the problem that much larger for evolution by adding reproduction. But we are going to engage that throughout this talk. If viral-like sequences in the genomes of organisms are functional, which we are going to demonstrate throughout this presentation, directly come from the conventional literature that endogenous retroviruses function in the immune system, they function in embryological development, they function in gene expression, they function in determining cell types, gene regulation, just to name a few of those functions, which we are going to cover in detail. But if viral fossils, which they like to say, are functional, why do they bear similarity to viral genetic material? Okay, you're going to see throughout this presentation that their very important functional and operational roles are dependent on their sequence similarity to viral genetic material. Some really fascinating information that we're going to be covering throughout this presentation. The next question. Why is the best explanation, or what is the best explanation for the nested hierarchical distribution of endogenous retroviral-like sequences in primate genomes? You're going to find out that it's due to design, reflective of design, not descent. For those wondering, what is a nested hierarchy? What do you mean by nested hierarchical patterns? Well, think of these groups within groups patterns. Okay, they'll say that nested hierarchies are essentially the reality or observation that humans are more similar to chimpanzees than they are to orangutans, than they are to monkeys, than they are to dogs, to fish, to bacteria. Okay, this nested hierarchy that we see largely in anatomy, that we see in genetics as well. Okay, and we are going to answer that question. And the last question, can separate ancestry explain the data better than common descent? The answer is yes, and we will find out exactly why. So stay tuned. We're going over a lot of important information here, guys. Endogenous retroviruses, the details. Let's cover the details, the important details. What are endogenous retroviruses? What are retroviruses in general? What is their genetic makeup? What is their structure? Okay, let's cover all of this before we show you exactly why ERVs are not good evidence for evolution. According to the standard evolutionary story of endogenous retroviruses, an ERV is a stretch of DNA found in your DNA that got there when one of your ancestors was infected by a retrovirus. That's why they will call ERVs fossil viruses, because what they believe is that millions of years ago, a primate-like ancestor was infected with an exogenous retrovirus. X means from outside, external infection. The most common exogenous retrovirus that you would be aware of is HIV. but HIV is passed on horizontally and not vertically. And you would get HIV from outside, external infection. That's an exogenous retrovirus. ERV means from within. You have ERV-like sequences in your body to start with, okay? And so they'll say that primate-like ancestors were hit with these retroviral infections, exogenous retroviral infections, but they infiltrated in the right way in the right spot, which would be in the germ line, okay, a reproductive cell. And therefore, rather than just being passed on horizontally, these are passed on vertically to the next generation, to progeny. Okay, let's continue. Fossil viruses. Proponents of common ancestry believe that ERV, the technical way of pronouncing it, or ERV, elements are the ancient remnants of past viral infections. Some evolutionists will refer to ERV sequences as fossil viruses. You can get all this directly from the conventional literature. We are, as a matter of fact, going to be dismantling ERVs and giving the true explanation for ERVs from the conventional literature, because many in the conventional literature that are putting out these truly incredible papers, they're not even realizing that the data that they are providing us, including some of their interpretations of the data, work towards overturning evolution and confirming creation. Let me say that again. Overturning evolution, confirming creation. We're refuting evidence for evolution, but we are also model building. We are offering a superior model, a superior explanation, and that would be the biblical model of ancestry. Notice this: human endogenous retroviruses, HERVs, when you see the H, it's human, represent, so this is their philosophy, this is what they believe, represent footprints of previous retroviral infection and have been termed fossil viruses. They are transmitted vertically, so this is that vertical transmission through the germ cell line, and are thus inherited by successive generations in a Mendelian fashion. So they're passed on to progeny, they spread. Okay, they would have to eventually reach fixation, become stuck in place. Let's move on here. Evolutionists acknowledge that for most of these ERV sequences, I say most because you're going to see that there are some unfixed ERVs in humans and other creatures, okay, where not everybody has them. And we are going to address why those exist. But for the majority of ERV-like sequences, the evolutionary community would argue, they would concede that these infections would have occurred millions of years ago. So they didn't see the infections take place. We're just seeing these ERV sequences in the genomes as they are. How'd they get there? Is the evolutionary explanation or story or philosophical ideas about how ERVs got there and ERV-like sequences, are those true? And we are going to see, we're going to engage that. Notice from this paper what they say. ERVs are proviral sequences that result from colonization of the host germline. So this is what we were saying earlier, germline by an exogenous retroviruses. So not endogenous, but exogenous. So externally, and it infects the germline and gets passed down horizontally? No, vertically. So there's that difference between horizontal transmission, vertical transmission. Notice what they admit here. The majority of ERVs represent defective retroviral copies. However, for most ERVs, endogenization occurred millions of years ago. The majority of viruses, okay, they don't infect the germline, which is a good thing. Could you imagine the HIV exogenous retrovirus infecting the germline, and now those effects, that virus is passed down to children and spreads that way? That wouldn't be good. Okay, but as the hypothesis goes, exogenous retroviruses that infiltrate the germline, they can be passed on vertically to progeny, to children. Occurred millions of years ago is what they say, obscuring the stages by which ERVs become defective and the changes in both virus and host important to the process. Okay, so here is a visual, an important visual to get you to understand what we're describing here in terms of horizontal transmission. Okay, you have somatic cells and germ cells. Somatic cell would be like a skin cell. Okay, so you get infected, it's not going to be passed down vertically. You're not going to get vertical transmission. So you get an exogenous retrovirus infecting, let's say HIV virus, then it can be passed on horizontally, okay, which the virus wants to be passed on, right? That's why you get cold viruses. They actually like the fact that you get sick, you get these symptoms, because then what do? You sneeze. And when you sneeze, you spread more viruses. And these viruses want to spread. Okay, that's their mission. And so HIV, the HIV virus would want to spread as well. Or you get, as you can see here, germ line infection, and that way you get vertical transmission. And so you get the infection here, and then it's passed on through reproduction to the children. And then from there, those people can pass on that DNA, and it spreads and eventually becomes fixed, is the hope. For evolutionists, they need fixation of beneficial mutations specifically to drive forward change. Okay, so here's our next visual. You'll notice that we have transposable elements. Okay, transposable elements are mobile genetic elements. Now, what this means is these sequences, these specific DNA sequences, they can actually change their position within the genome. They can move around. Okay. They make up just a truly massive portion of the genome, as you can see here in this visual. ERVs, or ERVs, which we're discussing here in this presentation, they're a type. So they're a very specific type of transposable element, specifically a subclass of retrotransposon. Okay, so that's why you've got 45% of the genome is made up of transposable elements. And then you've got retro elements, and you've got LTR, non-LTR retro elements, and your ERVs, which are your LTR retro elements. Different kinds of transposable elements here: signs, lines, short interspersed nuclear elements, long interspersed nuclear elements. What's interesting, what's amazing, is that all of these classes of retrotransposons, these various classes, they all have amazing functions. That we've discovered incredible functions for all of them. Okay. But you'll notice in these types, like signs and lines, okay, which are non-LTR, they don't have the long terminal repeat. Okay. But your endogenous retrovirus down here, you'll notice the makeup, LTR on either side. Again, that's a long terminal repeat. GAG, POL, ENV. Okay. So the perfect picture here, showing and covering the structure of the ERV. Okay. You've got the POL gene right here, and that encodes for enzymes like reverse transcriptase, protease, and integrase. Now, these enzymes are all required. They're needed by a retrovirus to do their job of what's called reverse transcription and then integration into host genomes, because retroviruses, they need to reverse transcribe their RNA into DNA. We know transcription as DNA to RNA to protein, right? Protein synthesis. So DNA transcribed into RNA, translated into a protein. Reverse transcription is simply just transcription, but in reverse. So instead of DNA to RNA, it's RNA into DNA. And it's important to understand the details of what a retrovirus is, because we are going to be getting into the origin of retroviruses in general. And so retroviruses are a special type of virus. There are different types of viruses. And the type that is a retrovirus, they have specifically an RNA genome. And what they can do through reverse transcription is actually convert they can convert that into DNA, the RNA, into DNA, and then insert it into the cell's genome. Okay. They can integrate it. And it was actually, if you go through the history of discovery, you will see that the enzyme reverse transcriptase, when that was first discovered, it revolutionized molecular biology. And this is because the central dogma was DNA to RNA. But now we have reverse transcription where it's RNA to DNA. And this amazing enzyme, reverse transcriptase, this enzyme of the retrovirus, okay, that's what is required. That's what converts the RNA into DNA. And again, a good way to remember what that is it's just transcription, but in reverse. And when the viral RNA gets converted into DNA, the DNA can now be read by the host machinery and then transcribed to produce viral RNA molecules. And so these components are necessary for, as we're going to see, the function of the ERVs, but also for retroviruses, because remember, retroviruses and ERVs share sequence similarity. And one of the questions that the evolutionist has, apologists and guardians of common descent, is why do ERVs share sequence similarity with retroviruses if they did not come from originally retroviruses? Now, the GAG, because again, you got the GAG, you got the pole, you got the ENV. The ENV is the envelope protein, okay, for the retrovirus, and the DNA element known as an ERV. The GAG gene codes for the core structural proteins of the virus. And again, it is your pole gene that codes for these enzymes like reverse transcriptase and integrase, protease. And so it's important to understand the makeup, the genetic makeup and structure of the ERV, because every single property, every single component of the ERV is necessary for many of their functional roles. Okay, so let's recap a little bit here, the details. How does viral DNA become endogenous? A retrovirus uses host DNA to integrate itself after converting their RNA genome into DNA via reverse transcription, and utilizes the cell's machinery to replicate. This is because retroviruses lack the ability to replicate themselves. This process leaves a copy of the retrovirus insertion in the DNA of the infected cell. If the infected cell is germline, so this is where you get the ERV, endogenous retrovirus, and the retroviral DNA is passed on, so vertically rather than horizontally, every copy of the descendant's DNA will have this viral DNA. And now that we understand the details, the important details, we have set the foundation for answering many of these questions. Here's the first major question in challenge that I want to interact with. Why do endogenous retroviruses share sequence similarity to exogenous retroviruses? And you are going to see here that ERVs have numerous incredible functions, but two of those functions are tumor suppression and acting as antiviral protectors. And when it comes to tumor suppression, what their function is viral mimicry. And in order to carry out that function, they actually have to share sequence similarity to a retrovirus in order to carry out that function, because it has to look like that which it is mimicking. So it has to have the LTRs, the GAG, the pole, the pro, the ENV components. And acting as antiviral protectors, they actually fight off harmful viruses. In many ways, they are our system defenders, and they are akin to how in your computers, you have antiviral programs. You might have Norton antivirus or Windows Defenders or whatever. And those fight off or protect your computer system from harmful viruses and invaders, and so do ERVs. But we're going to get into those in detail. Important functions. Numerous functions have been identified for endogenous retroviruses. So again, ERVs are playing an antiviral role in the genome. And to carry out that role, again, they got to look like the virus because their components, okay, like the proteins, for example, the ERV proteins, they actually interfere with incoming viral particles. The proteins encoded by the ERV units in the genome, they heighten or amplify immune responses, and they do this through the binding of very specific receptors. Okay, and so the makeup, the properties of the ERV, they are what help to carry out these functions. They have to share sequence similarity to retroviruses to in order to carry out these roles. For example, the envelope proteins, they function in blocking harmful viruses from entering. This is amazing. This is evidence of forward thinking, which points us back to what? To who? The forward thinker. And these ENV proteins, they do this by binding to receptors that infectious viruses might use to enter the cells of hosts. So there's a lot going on in terms of how they carry out these operational roles. And these ENV proteins, they have the potential to actually enter viruses. And as a result, they disrupt the viral life cycle. And this whole process inactivates viruses before they can infect new host cells. This is amazing design. And again, this is forward thinking. Here's a couple other functions. Determining cell types and also regulating gene expression. Now, gene expression, gene regulation, very important to the cell. Gene regulation is essentially that process used to control the timing, the location, and amount in which genes are expressed. And then gene expression is that process by which a gene is turned on to produce an RNA or a protein. It's this process by which DNA directs the synthesis of proteins, protein synthesis. Okay. And gene regulation is regulating this process. And you can think of eukaryotic gene expression in this way. A good illustration to understand it better is a stoplight. You're driving your car, you come to a stoplight, you've got your red light, you've got your yellow light, and your green light. Okay. And so for genes, they can either be turned on. That would be represented by your green light. That's a gene that's on. A gene can be turned off. That is represented by your red light. Or a gene can be expressed at a low level expression. That would be your yellow light, represented as your yellow light. And so these ERVs are assisting. They're functional in these very significant processes. Okay. Again, gene regulation, gene expression, determining cell types. ERVs are essential in regulating early stages of human embryonic development. I want you guys to pay very close attention to this paper right here. Okay, because they admit directly in the conventional literature that if we didn't have ERVs and ERV-like sequences, we couldn't exist. I like to exist. The evolutionist that uses this argument as a piece of evidence for common descent, I hope they like to exist. The viewer likes to exist. We wouldn't exist without ERVs. Notice this. As we have summarized in this review, HERV, so there's that H again, that means it's a human ERV. They appear to play important roles in physiology, fetal development, and human evolution. Now, they always have their human evolution biases. They always have to take the data and put a human evolution spin on it, right? What that means, human evolution, is basically the fact that ERV-like sequences, they actually help to promote genetic variation and create diversity, which is what we'd expect from the design diversity model. Okay. And so here is a very important excerpt from this paper. And this is discussed in many papers dealing with the functions of ERVs. If the accidental infection of a mammalian ancestor by an exogenous retrovirus had never occurred, the placenta and the mammals that produce it, including humans, notice this, would have never existed. Never existed. Why do we share ERVs with chimpanzees? Why do we share ERVs with primates? Okay, with all kinds of creatures in the biological world. Well, why do all modes of transportation have engines? Why do they share many engines? They have engines in common. Well, without an engine in your vehicle, you're probably not going to get very far. You're not going to be able to use your vehicle for what it was designed and engineered and intended to be used for. Okay. And for mammals to reproduce, for the placenta to exist and to work, we require ERVs. And so these are required for life. This is good design. Chimpanzees, orangutans, gorillas, other primates, they have babies. These processes are very similar. Okay. And so in the early stages of development, what we have is the development of a placenta. And to give a little more detail, a placenta is a temporary organ that forms during pregnancy. Okay. And this structure is crucial. It is essential for normal and healthy pregnancy. It provides nutrition to the developing baby, and it also helps to get rid of waste. So it's this cycling process of waste and nutrients. Very important. And for the placenta to work, this structure needs to be connected to the baby for these processes to be able to take place. Okay. And it turns out that an endogenous retrovirus is key to this process, to embryological development and placental function. The placenta actually secretes a protein that binds it to the embryo, and that keeps the two attached for the next several months of development. And the DNA that makes up that protein is very similar to a region of a retrovirus that allows the virus to attach to its host cell. Okay, so the structure and design of this ERV is necessary for what needs to take place in embryological development. Guys, without endogenous retroviruses, the placenta, which again is critical to pregnancy and embryological development, would not work. We essentially would not exist. These functional DNA elements are essential for life. And therefore, of course, we're going to share them with the biological world. Here's a couple papers. Additional, and notice this is all from the conventional literature. We are dismantling their so-called best evidence directly from the conventional literature. Scientists identify new beneficial function of endogenous retroviruses in what? Immune response. Endogenous retroviruses function as gene expression regulatory elements during mammalian pre-implantation embryo development. Where did the evolutionary community predict this level of functionality in what they call fossil viruses or fossil sequences? That's what I want to see. Beneficial role of human endogenous retroviruses, facts and hypotheses. HERVs have recently been suggested as mediators of normal biological processes, such as cellular differentiation and regulation of gene expression. Now, cell differentiation, what that is it's basically going from unspecialized cells to specialized cells forming tissues and structures and functions. And ERVs have a role in that, in cell differentiation. And so again, the functions are in abundance. It's not just in the placenta and embryological development. Okay. The ERV and ERV-like sequences also work in our immune systems, as we've seen. And our immune systems are these pathways in our immune systems. They're dependent upon enzymes created by endogenous retroviruses and ERV-like elements. ERVs also safeguard the growing baby in the womb during pregnancy from harmful viruses and invaders from coming in. So a lot of ERVs are only functional under certain stages, and then they're shut off. So they do their job and then they're turned off. And evolutionists, after the ERV is done doing its job and it's turned off, they could do a knockout experiment, knock out the ERV, and it has no effect to phenotype, okay, to the organism. And they would conclude, based on evolutionary assumptions, that this ERV is non-functional. No, again, a lot of ERVs, a lot of what the evolutionary community refer to as fossil viruses, evolutionary leftovers, they're only functional during very specific stages of development, or only functional under specific in specific environments. Okay, so there is a lot going on here in terms of what ERV and ERV-like sequences are doing. They're involved in allowing for small scale but very important changes in how the human body works in general. What does this mean? That yes, they should be shared among all kinds of life in the same way that batteries and engines and all types of important features of vehicles are shared. This is good design. This is what a creationist would expect. Okay, the design hypothesis would suggest this. We start with the Bible. The Bible claims to be the history book of the universe. And the Bible tells us in Genesis that we are created in the image of God. What's the hypothesis, therefore? Well, there must be something about us that reflects the divine. Okay, so perhaps we can get a sense for how God created based on the way we created in the image of God as human engineers and human designers design and build things. It just so turns out that we design in homologous patterns. You can take vehicles from North America, Asia, and Europe. Okay, so you can take a Chrysler, you can take a Honda, and take a Volkswagen. And you're going to find all kinds of features in common. That doesn't mean that they inherited those features from a common vehicle ancestor in the Arctic. Humans build in homologous patterns, and it turns out that the biological world is created in homologous patterns. We share ERVs with other creatures. That's good design. Human engineers also, by accident, they're not trying to help confirm the biblical creation model. Human engineers also design in what? Nested hierarchical patterns. A sedan shares more with a hatchback than it does with a boat or an airplane. Okay. A sedan and a hatchback have more in common than they do with a semi-tractor trailer. So you have these nested hierarchical patterns in design modes of transportation, and you also have nested hierarchical patterns in the biological world where humans look more similar to a chimpanzee than they do to a dog or a fish. And so, of course, these functional created units of DNA like ERVs Obviously, we look more like the chimpanzee. We have a lot more in common with the chimpanzee. Therefore, we are going to share more of these ERV sequences and ERV-like sequences with the chimpanzee than we do with a dog. Okay. And so the nested hierarchical distribution of ERV and ERV-like sequences in the biological world is really not a huge surprise. And moving on, this is one of my favorite papers. This paper covers it all. So if you're listening to this right now, save this paper and read through it extensively. It is an important one. Notice this: switching sides, how endogenous retroviruses protect us from viral infections. Long disregarded as junk DNA. So they admit that they assumed ERVs are junk DNA. Where did they predict this large scale function? And I'm talking about large scale function. Where did they predict this prior to the discovery? They often talk about in the secular literature that they're surprised, they're caught off guard, amazingly. Okay. Long disregarded as junk DNA or genomic dark matter, ERVs have turned out to represent important components of the antiviral immune response. They not only regulate cellular immune activation, but may even directly target invading viral pathogens. That's what we were discussing earlier. The viral proteins, the components and properties of the ERV, they actually act to heighten and strengthen immune responses to incoming viral invaders. They help to stop the infectious process. This is amazing stuff that's going on the molecular level. One focus will be on recent advances in the role of ERVs as regulators of antiviral gene expression. And so what's the overall answer to ERVs? Well, these DNA sequences, and here's the thing to consider. The ERV did not call itself an ERV. It was named based on the assumption that, hey, these are the ancient remnants of past viral infections. That's why the question at the beginning, are these really the ancient remnants of past viral infections, or are they created units of DNA function? I think we're seeing very clearly, and any objective thinking person listening to this and seeing this data and seeing these papers, they have to admit that the better explanation or even name for these DNA units are created units of DNA function. Designed units is what we are looking at here. And these were front loaded in the beginning at creation by God in living organisms as design units of DNA that operate in many ways. One major way is here in the immune system. And the fact that they resemble viral genetic material is because these similarities are necessary for their functional roles in interacting with real retroviral DNA. And these roles are amplified during immune responses. This paper in detail goes over these functions. And before we move on, though, you'll find a lot in the evolutionary community, they'll just assert co-option. Okay, they'll just assert that, well, these functions were co-opted. And what they'll say in terms of co-option is that components of the ERV, right? The ERV is made up of what? LTRs, GAG, POL, ENV genes. So they'll say, well, these can be co-opted by the host organism. Okay, now that the retroviral DNA is in the host is like, well, we might as well utilize this DNA and these components. And these properties can be repurposed for other things, for other functions like placental development or functions in the immune response. Okay, but they will oftentimes just assert co-option with no real evidence. Okay, are ERVs going to be co-opted, or are they going to evolve a beneficial mutation? The answer is largely no, because randomly inserting viral DNA into the genome of a biological organism will more likely disrupt genetic functionality rather than improve upon it. These types of changes are usually functionally compromising to biological organisms. If I was a painter and I was painting a beautiful picture and then I applied a blindfold, okay, over my eyes, and I just started randomly applying brush strokes. Am I going to improve the painting or make it worse? I'm probably going to make it worse. And so that's their philosophy. They can believe that these functions came about through co-option and beneficial mutation accumulation, but they really do lack the evidence for that. They have never observed in a lab or in real time an exogenous retrovirus infiltrating, infecting, being passed on through the germline, and then mutations wipe away the harmful effects of the ERV provirus, and then eventually they evolve a novel function. They haven't observed this. If they have, show me. And show me it happening in something essential like embryological development. Another important point to note, because the apologists of evolution, they are frequently guilty of not giving the full picture of what we're dealing with. They'll say, oh, we share all of these, just thousands and thousands of ERVs with chimpanzees and primates and other creatures. But what they fail to tell you is that only 10%, consider this, guys, only 10% of ERVs represent full ERVs. Full ERVs being what we went over earlier. Do you see why the details are important in addressing ERVs? The LTRs, GAG, POL, ENV, only 10% actually represent full ERVs. Over 90% of ERVs represent solo LTRs. Again, let me restate. Over 90% of ERVs, they don't even have your GAG, your POL, or your ENV. They're just solo LTRs that evolutionists are assuming are degenerated ERVs. Okay. And long ago and far away, they at one point were a full ERV. Okay, no, they don't even resemble an ERV because they lack the GAG, the POL, the ENV components. They simply represent solo LTRs, long terminal repeats. And I would argue that they're just functional stretches of DNA. Oh, but Donny, how can you just assert that these are just functional stretches of DNA? Therefore, common design being the better explanation for why we have thousands of solo LTRs shared in the biological world. Well, that is because solo LTRs are serving functions. They're serving as binding sites for initiating and enhancing gene expression. Okay. This tells us that your full ERVs and your solo LTRs, which are assumed to be degenerated ERVs, they all have amazing functions, very important and essential functional roles. And therefore, the question is this: are these solo LTRs degenerated ERVs or functional stretches of DNA? What does the literature say? Guys, today, what are we doing? We're debunking ERVs directly from the conventional literature. Isn't it a great time to be a biblical creationist? And it's not such a great time to be an evolutionist. And by evolutionists, we mean somebody who believes they're related to whales and pine trees and banana plants and strawberries and chimpanzees because, well, we share ERVs. Notice this. They admit exactly what I just said. Almost 90% of all HERVs represent so-called solo LTRs. These HERVs lost the prototypical retroviral genes GAG, POL, and ENV due to homologous recombination of their flanking LTR sequences, leaving single LTR promoters in the genome. LTR, they're a good tool. They're a good building block or functional unit. Okay, just like we have certain units or tools that can be used throughout many modes of transportation and used in different ways, in a variety of ways. So we have an LTR unit, okay, which is functional. They act as promoters. They serve in initiating gene expression, right? They serve as binding sites, all these different functions, okay? And so God can use these units, these LTR units, in many different ways in the genome, just in independent positions, They're utilized in ERVs. Retroviruses have LTRs. There's nothing wrong with a functional unit that is used in a variety of ways. And yet we have so many of these large-scale function for these solo LTRs, and the evolutionist is going to assert without evidence that, well, these just happen to land in the right spot or be in the right spot or be integrated in the right way, very intricately, to serve in these functional ways, hundreds to thousands of times. Now, this is their philosophy. They can believe it. Okay, they can even yell it loudly and proudly, but that's their philosophy. It's not science. The science contradicts their story of endogenous retroviruses, leaving single LTR promoters in the genome. Notice this. And evidence. Let's go through the context. Because I had one evolutionist make a video saying, oh, LTRs, how do they contribute to immune system responses? Due to their activation upon immune stimulation, ERV LTRs have already been termed landing strips for inflammatory transcription factors, and evidence for their role in regulating cellular immune responses is growing. So these evolutionists that say, well, how do the LTRs apply to immune system responses and acting, in protection in the genome and in the immune system? Well, what does it say here? Evidence for their role in regulating cell immune responses is growing. It's amazing. The endogenous LTRs mentioned above all act as enhancers and may increase, let me say it again, increase antiviral gene expression in response to viral infection and or IFN stimulation. I'm going to be putting out a video response to a population geneticist, Dr. Zach Hancock, going point by point through his video. And you guys, it's important. Remember what we're saying here on the LTRs, because these points directly from the conventional literature work to counter a lot of what he says. Here's a couple more papers. Screenshot this, save these papers, go read them for yourselves. Okay. Don't just believe what I'm saying without looking into it. Look into it and you're going to see what I'm saying is true. Endogenous retroviral LTRs as promoters for human genes. Endogenous retrovirus long terminal repeats as ready-to-use mobile promoters. ERVs can be reactivated by viral infections. While many of them are usually dormant, they can be reactivated by several stimuli, including viral infections. Isn't that amazing that they can be dormant? They are silent until they need to be utilized, until they need to be called upon to do their job. ERVs have become integral parts of immune defense mechanisms. Notice how they say have become, or perhaps they were created that way, designed that way, as functional components in immune defense. They'll never think that all of this, they've got their evolutionary bias, evolutionary starting. They won't question the bigger picture. They can question details of the bigger picture, but not the bigger picture itself, and help to fight off invading viral pathogens. Interference. This is what I was describing earlier. And what do we have here? Directly from the conventional literature. Apart from rather broad and unspecific immune modulatory activities, endogenous retroviral ENV proteins can also directly inhibit viral infection through a process termed receptor interference. These evolutionists, these desperate evolutionists, including PhDs, that say, well, it's not that the ERVs are functional. It's just that there's certain parts of them that are functional. ERV itself. These guys are out of date and out of touch of reality. Okay. Everything about the ERV is functional and necessary to sustain healthy life processes in the cell. The ENV, the GAG, the POL, everything about the ERV, the LTRs, they're all functional. It's not, well, just this part of the, no, it's all functional, all of it. And the arguments, the criticisms, the so-called rebuttals that I have seen over the years, they only prove that the evolutionary community, apologists of common descent, they're out of date on their own favorite argument. And listening to what these guys have to say on ERVs or in response, I was sent a video where Forrest Velkai and Godless Engineer attempted to engage ERVs, and it was just dodge, duck, dip, dive, and dodge. The 5 Ds of dodgeball. They addressed nothing. And I sat there and I thought, wow, these evolutionists, even the most militant of evolutionists, they really got nothing. Okay. And that's why they won't actually engage what's being said here. They're just going to repeat and parrot their talking points, because in reality, shared endogenous retroviral-like sequences in the biological world, they're on the side of the creationist. The so-called best evidence for common descent is now some of the best evidence for creation and for separate ancestry. Notice this: since exogenous and endogenous retroviruses frequently share the same receptors, they have to share the same receptors. Endogenous ENVs, that's the envelope protein of the ERV, offer a large repertoire for broadly active antiviral factors. They have to share sequence similarity with the retrovirus to carry out many of their functional roles. Another one of them, these roles that I didn't go into in very much detail yet, is the viral mimicry. Okay, transcribed ERVs, they give the cell the appearance that it's being invaded, okay, and by a harmful retrovirus. And so the immune system, working together with the p53 protein, which is the guardian of the genome, this incredibly designed and essential protein, the p53 protein, they work in combination. The immune system targets that cell with more destruction, killing tumor cells. So you have a tumor cell, the ERV through viral mimicry, the immune system, the p53, all these amazing things are going on. These tumor cells are being destroyed. But guess what? If the ERV didn't look like the retrovirus that it's mimicking, it wouldn't be able to carry out that amazing functional role. So when an evolutionist asks, and oftentimes in a rhetorical manner, why do ERVs look like retroviruses? If they didn't come from retroviruses, you know that they didn't read any of these papers. They're out of date. They're on first base. We're on third base. Okay, they're way behind us. That's why a lecture like this is important, because it is the definitive talk on ERVs that you can study, examine, watch, pass around to your friends, your family, skeptics, critics. The next time a critic brings up ERVs as so-called evidence for evolution, send them this video. Tell them to refute it. Challenge them to interact with these points. And for the most part, they won't. It would be like an FBI agent infiltrating the mob, and he shows up to infiltrate the mob, pretending to be a mobster, and he shows up in his FBI uniform with his badge. Hey, my name's this. I'm from the FBI. He's not going to get very far in his job, is he? No, he'd have to play the part. He'd have to look the role. And so the ERV has to look the role. I like to say paper after paper. We're not going to have time to go through every single one of these papers. If you pick up a copy of my handbook on the topic, I go through and cite all these different papers that you can enjoy and study and read through. And so here's a few. Retroviral promoters in the human genome. Endogenous non-retroviral RNA virus elements, evidence of a what? Novel type of antiviral immunity. Notice what they admit in the conventional literature, guys. Increasing knowledge. Our knowledge about endogenous retroviral elements involved in cell immune responses is constantly increasing. ENVs, recent key findings include the discovery of retroviral ENVs restricting retroviral infection in primates. I wanted to skip over to a separate presentation that I have here. I've got hundreds of slides on this topic and other topics. And there's so much to dig into from these papers. This goes back to the switching sides paper. And notice this. Early reports of receptor interference. These date back to the 1960s. Looking at these ENV proteins in avian leukosis viruses induce what? Resistance to exogenous retroviruses. These are all important in immune responses, guys. Besides ENV, endogenous retroviral gag proteins. So these guys, these evolutionists that want to say, oh, it's just a part of the ERV. It's not the ERV that's functional, it's just a part of it. Something, part of it was co-opted. They've got all these different kinds of worthless excuses. No, every single aspect of the ERV, the gag, the pole, the ENV, the LTRs on either side, they're all important. They're all significant. Besides ENV, endogenous retroviral gag proteins can also restrict retroviral replication. We read earlier the LTR is involved in immune system responses. Thus, the ability of endogenous viral gag proteins to interact with their exogenous counterparts may confer blocks at several steps of the retroviral replication cycle. What about transcriptionally silent ERVs? Those must be non-functional. That must be evidence for common descent. No. In the conventional literature, we now know that transcriptionally silent ERVs, transcriptionally silent pseudogenes, they're functional in many ways. One way here, transcriptionally silent ERVs can also provide a selection advantage to the host. For example, the presence of repetitive ERV elements may promote recombination. These recombination processes that increase the number and or allelic variety of host genes. Okay, just look up the number of papers, type in, do some basic research, transcriptionally silent ERVs and transcriptionally silent pseudogenes. What are their functions? What have they discovered? And they're highly functional as well. It's not looking good for the evolutionary community. And that's why it is so important to stay up to date on the literature, because nobody wants to be using outdated arguments. In the human genome, these HERVs, okay, HERV-H, for example, strongly influences our growth, development, and our genetic code. Well, thank God we got these ERVs, okay? And in koalas, there's a paper, and I cite it in my book. I've cited it in other videos. In koalas, they are experiencing retroviral infections, and some of them are being passed on vertically, becoming endogenous retroviruses. Well, it turns out that the pre-existing endogenous retroviruses, they're actually working to disrupt the endogenization process of these new endogenous retroviruses. Everything going on in terms of the attacks on the koalas, your pre-existing ERVs, which I would say, according to my model, are created ERVs. They're working to protect the koalas from these retroviral infections and the subsequent endogenization process. Okay, so there are just a number of fascinating roles for these ERV and ERV-like sequences. As I like to say, paper after paper. Transposable elements have had a substantial and sometimes beneficial impact on host genomes in several ways. You can have a look at all these different papers here that I have listed. Important in gene regulation in early mouse development. There's a specific class of retrotransposon in the mouse, and they snip it out. The mouse is developing, they snip it out, and it stops developing. It dies. Because all kinds of creatures, their developmental process depends on retrotransposons. TEs contribute to transcription regulation endogenous retroviruses are emerging as promising therapeutic targets in cancer. This is what I was talking about earlier. There's several papers. I was just doing some more research on this last night because I'd like to do just a whole separate video on viral mimicry and how ERVs perform viral mimicry and works with the p53 protein and also the immune system to destroy tumor cells. Far from being junk DNA, the pervasive retrotransposons that populate the genome have a what? A powerful, guys, notice the language, a powerful capacity to influence genes and chromatin. When you see the word chromatin, that's referring to a mixture of DNA and proteins that form the chromosomes found in the cells of humans and other higher organisms. And what are these retrotransposons doing? They've got a powerful capacity to influence genes and chromatin. HERV-H, okay, I mentioned HERV-H just a second ago, can modify the higher order 3D structure of chromatin during early primate development. Another amazing function of HERV-H, okay, so human endogenous retrovirus, for a short time during embryological development, embryonic cells are what's called pluripotent. Okay. What this means is that they can become multiple different types of cells. And our bodies require certain types of cells in certain places for normal development. This ensures our heart, our muscles, brains, lungs, okay, they end up developing in the appropriate spots. This is very important for normal development and growth. Now, instructions in the genome told the cells what kind of tissues to permanently become as they become more specialized. HERV-H, right here in this paper, that says can modify the higher order 3D structure of chromatin during early primate development. HERV-H marked our cells as pluripotent and ensured that they remain in that state. This would prevent them from turning into muscle or skin cells for just a little bit longer than they would otherwise. Do you see the amazing control that these ERVs have? Okay, again, these types of functions point us back to the forward thinker that front-loaded organisms with these DNA elements, these incredible DNA elements. And so when it comes to the HERV-H and pluripotency, the HERV-H, it keeps these cells pluripotent for longer. And that means those genes are expressed longer as well. Okay, so this so-called ancient viral fossil has a massive impact on embryological development. HERV-H has a role in shaping heart development. Our bodies really would not be the same without these ERVs. We wouldn't exist. HERV-K, that's another human endogenous retrovirus, helps embryos develop a built-in immune system that keeps them safe even before they develop antibodies to pathogens in the outside world. Okay, so in addition to all the numerous functions that I've covered in this lecture, I know I've covered a lot. I know this has been technical. It's comprehensive. That's why I called this the definitive talk on ERVs. One talk, one video, one presentation that you can go to, and you can basically get all the necessary information to completely dismantle ERVs, but also understand the details of ERVs and retroviruses in general. And so in addition to these functions that I've covered in great detail in this lecture, endogenous retroviruses even positively affect our pluripotency. They help our immunity as an embryo. They also play a major role in making sure that we develop into a fully grown baby. It should not be a surprise that we share endogenous retroviruses or that there are nested hierarchical patterns in endogenous retroviruses and where they're located in the biological world. Notice what they say here. Endogenous retroviruses are an unexpected ally in the regulation of human immune systems. That key word, unexpected. Inconsistencies with even though there's a general hierarchy, when you look at the details, when you look at the specifics, specific cases, you find that there are many inconsistencies. And we've done whole videos on inconsistencies in the nested hierarchy. This lecture is not really aimed for that or designed for that, but there's convergent evolution. Anytime you see convergent evolution invoked, the evolutionist is simply just admitting to a lack of uniqueness in the universal phylogenetic tree of life. Okay. There's incomplete lineage sorting, where depending on what you look at in terms of a gene or a genetic marker, you get totally different trees. Here's an interesting one. A HERV-K provirus in chimpanzees, bonobos, and gorillas, but not humans. Well, that's interesting. These observations provide very strong evidence that for some fraction of the genome, chimpanzees, bonobos, and gorillas are more closely related to each other than they are to humans. I like to talk about the Y chromosome, right? The Y chromosome dissimilarity within the great apes and humans. That when you consider overall architecture, size differences, and gene content, your human Y is more similar to your gorilla Y than either are to the chimpanzee Y. There's a break in the so-called nested hierarchy. So we could talk about inconsistencies all day. But yes, there is a general hierarchy, but this is a creationist expectation. Of course, we're going to share more of these functional DNA units with the chimpanzee than we would with a dog or a fish. Just stand afar. We look more similar to a chimp than we do to a dog. And so if the design hypothesis regarding ERVs is true, then we have an elegant explanation for the nested hierarchical distribution of ERVs. And so, guys, that wraps up part one, because I do want to dedicate as much time as is needed. And we've already covered just a ton in this video. And so part two is just going to be a superior model. I am going to show you the superior model in terms of explaining the origin of ERVs, the origin of retroviruses, and showing why there's really just an unanswerable chicken and egg problem for the evolutionary model. I want to address some criticisms that I've seen pop up. And that'll probably take me another 30 minutes to 60 minutes. And so I want to leave this video where it is, going over all the functional roles, going over the reasons for homology and nested hierarchies, showing why the evolutionary community really can't account for the functional nature of ERVs and ERV-like sequences. And then the next video, which I'm hoping to get out sometime within the next few days, as I've got it all ready to present. All I got to do is present it, record it, and upload it. We'll see part two very soon. And I think you guys are really going to be fascinated with what I have to present in terms of the superior model, because what I have to say, I truly believe is a major game changer. It also works towards explaining in even more of a powerful manner as to why retroviruses and ERVs look alike, share sequence similarity. And this is going to explain the origin of harmful retroviruses in general. Why do they exist? Why do they cause disease? So stay tuned for that. Again, I want to wrap this up here just to give you this video covering everything that we've talked about. And then the separate video is going to be strictly describing the superior model And I got to say, I'm excited for you guys to see this information because it's backed up. It's because I've done a lot of research in terms of the superior model, the creationist model, and a lot of what I say, a lot of the conclusions I make are backed up directly from the conventional literature and from many lectures from virologists and those in the evolutionist camp. So stay tuned for that. I hope you guys found this helpful. Again, this was the definitive talk on ERVs or endogenous retroviruses, the so-called best evidence for evolution. And we've shown exactly why ERVs are no longer a good line of evidence for evolution. This is why I like to say the best so-called evidence for evolution ends up becoming some of the best evidence for biblical creation. Guys, it is truly an amazing time to be a biblical creationist. God bless you all.